In the Caldwell lab, aka the Worm Shack, we study neurological disease mechanisms in C. elegans. And after a long day of fruitful experimentation, we reward ourselves with some recreational activity.
Hi, I'm Adam Knight, an undergraduate student from the Caldwell Laboratory in the Department of Biological Sciences at the University of Alabama. Today, myself and others will show how to perform a dopamine neuron degeneration assay in C. elegans. I will be crossing a dopamine neurodegenerative worm strain with transgenic strains that overexpress candidate disease-related genes in dopamine neurons.
Hi, I'm Adam Harrington, a graduate student in the Caldwell Lab. Today I will be showing you how to set up the transgenic worms for dopamine neuron analysis.
Hi, I'm Hamamichi, and I'm a graduate student here. I'll be showing you how to assay anterior dopamine neurons of C. elegans, which are called CEPs and ADEs. Generally, the worms expressing only GFP in the dopamine neurons do not exhibit cell death. However, in presence of alpha-synuclein, you will observe dose and age-dependent neurodegeneration. Using this assay, a transgene could be co-expressed in the dopamine neurons to determine whether it can enhance or suppress alpha-synuclein-induced toxicity.
So let's get started. Before carrying out the dopamine neuron analysis, we must first cross the appropriate C. elegans strains. To begin, place 8 males expressing both alpha-synuclein and GFP under the control of the dopamine neuron-specific promoter